In this video, we will discuss about this Oncolytic Herpes Simplex virus treated PSC, that is pancreatic stellate cells, can activate T cells and helps in neutrophil migration. So far in the previous video, we have seen that this oHSV treated cells can cause more antigen presenting CAF expression and less myofibroblast, which is a tumor proliferation CAF suppressions. So they wanted to check whether this oHSV treated PSC can directly activate the T cells and can help in this neutrophil migrations or not. So in order to check that, they co-culture the CD8 plus T cells and CD4 plus T cells with PSC treated with Oncolytic Herpes Simplex virus and PSC without treated with Oncolytic Herpes Simplex virus. And what they are able to find out that when this CD8 plus T cells, when they are co-cultured with PSCs which are not treated with Oncolytic Herpes Simplex virus, there is not much significant increase. However, when they co-cultured these PSCs that are treated with oHSVs, they are able to see a significant increase, that is 7.85 from 4%, almost like 1.5 times increase. That can be seen, the quantification results can be seen in this panel B, that CD8 plus T cells increase significantly when they are treated with this oHSV. And similar results were seen for CD4 plus T cells, that is when they are co-cultured with PSC without oHSV treatment, they see a very small increase. However, when the CD4 plus T cells co-cultured with PSC along with this oHSV treatment, they are able to see a significant increase, and the quantification results are shown in this panel D. So from these results, it is clear that CD8 plus and CD4 plus both T cells will increase when they are co-cultured with PSCs and Oncolytic Herpes Simplex virus. So indicating that the Oncolytic Herpes Simplex virus helps in this T cells activations. Thereafter, they also checked one factors that helps in the tumor proliferations or immune cell suppressions, that is regulatory T cells. And the regulatory T cells, they are able to see the similar, they are able to see the expected results, we can say that. For example, this CD4 plus T cells with PSCs shows increase in this this regulatory T cells to some extent. However, when the CD4 plus T cells are treated with this oHSV, when the CD4 plus T cells and co-cultured with this oHSV treated PSC, they are able to see a decrease in the regulatory T cells to almost about half. These things they have quantified in this panel lab, where we can see that the regulatory T cells suppressed significantly compared to its control, indicating that this Oncolytic Herpes Simplex virus not only helps in the increase in this antigen presenting markers, but it will also suppress the tumor proliferation markers or immune suppression markers, we can say that. So afterwards, they also wanted to check the effect of this Oncolytic Herpes Simplex virus on the neutrophil migrations. So in order to check that, they did the transwell assay, where they culture the lower chamber with these pancreatic stellate cells and then treat them with this Oncolytic Herpes Simplex virus. After 24 hours of treatment, after 24 hours of the oHSV treatment, what they did is they add the neutrophils that are isolated from the blood of this peripheral blood of this immunocompetent mice. And after 24 hours, they detect the neutrophil migrations in the in the bottom chamber with and without oHSV treated PSCs using flow cytometer and ELISA. And the result shows that the neutrophil migrations is significantly higher in the oHSV treatment. The panel H shows the result of this percentage cells, and panel I shows the result of the actual number of cells. Both of these shows that the neutrophils migration significantly increases when they are co-cultured with this Oncolytic Herpes Simplex virus treated PSCs. Along with that, they also checked this TNF-alpha, and TNF-alpha signals also significantly upregulated. So this all these results along with the previous results now pointing out come to conclusions with these results, and also the results that are presented in the previous figures. They come to a point that is oncolytic herpes simplex virus helps in this antigen presenting, helps in the upregulation of this antigen presenting cancer-associated fibroblast, thereby helps in this T cells activations and immune cells migrations, especially neutrophil migrations. At the same time, they will also suppress the tumor proliferating markers such as regulatory T cells and myofibroblast. This is what they finally conclude from this paper. I hope you understood the overall picture of this paper and the results that are presented in the individual figures. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.